Thank you to Adrian and the organizers for the invitation, and thank you guys for staying through this session with many speakers. One of the things which I think is really terrific about this meeting is just how incredibly diverse this audience has been. We've listened from people who are pure computational to many of you who are clinicians to ethicists this morning, and I'd like to make a case for representing the basic science researchers today. So just to start, I don't have any financial disclosures. This work, as Sarah was describing, was work that I had done during my postdoc at MIT in the Broad Institute. And toward the end, I'm going to share with you some of our vision for where we're going to go forward with this. I think it might be helpful. We've covered a lot of topics related to how AI and machine learning may be able to augment the ways in which we perform clinical care. But I thought it might be useful for us to maybe take a step back and think about our healthcare systems as a whole and where some of the intervention points are. And I think that I live in the States, but I imagine that here in Switzerland it's not too different. Oftentimes, a patient comes into a clinic because they're symptomatic, and then there's some sort of a series of events occur in which a diagnosis is performed, and that leads to selection for some sort of treatment. And many of the examples that we've been talking about over the last 2 days, really, we've described applications for machine learning and artificial intelligence, really at the task of kind of improving our ability to make appropriate diagnoses, or alternatively, in the direction of where I think there's a lot of aspiration with precision medicine to enable us to make better therapeutic decisions, whether it's because maybe you have some patient variability or maybe there's something special about the ailment that we try and treat. And for me, as someone who actually trained as an engineer, one of the things that I'm drawn to is trying to understand, where else can we find opportunities that might be very impactful. And so what I'm going to talk about with you about today is some of our ideas on how we think that AI can do more than just improve our diagnostic ability or more than just help us better stratify patients, but thinking about like what can AI do so that, or what can machine learning do so that we can synthesize new therapies that maybe don't yet exist today, or maybe even perform the harder task of learning strategies for preventing diseases. I'd be remiss if I didn't comment that there is a tremendous amount of activity right now in using machine learning to actually do proactive drug discovery, and point you to a paper that was recently accepted by one of my former colleagues. It's going to be published in the next couple of months in Cell, where what they've done is they've used deep learning to mine several different antibiotic resources to discover new antibiotic compounds that they've been able to show is efficacious against a few different pathogens. But instead, what I'm going to be talking about today is more from the basic science side, which is if we wanted to come up with new therapies, if we wanted to rationally come up with new therapies, how do we do that? And I think that one of the necessary prerequisites for rational design of therapies is at a fundamental level, having a really basic causal mechanistic understanding of whatever sort of phenomena that we're trying to study. And this is something that I think is actually quite hard. I think most of us can appreciate that, machines are really good at doing sort of association type of activities, but it's really hard, really difficult to infer, the sequence of events or the mechanisms that are really driving the clinical or cellular phenotype that we can see. There are some efforts right now, being taken by several different groups to try to address this problem. So one example of these, which I think was probably one of the earliest demonstrations, and this relates to, I think, Claire's talk yesterday about ontologies, is an approach where instead of using a completely completely unsupervised deep learning, a deep neural network approach, folks from the Trey Ideker lab describe an approach where perhaps what we can do is we can use a structure that's encoded in some sort of ontology as the network that we use for interpreting our data sets. And he was able to demonstrate that in analyzing yeast data, they were able to predict growth phenotypes for several different perturbations. I think amongst the computer scientists and amongst the computational people, I think the alternative approach, which has been really popular and kind of in part by this book that was published a couple of years ago, by Judea Pearl, is to think about designing our algorithms in a way in which that we use maybe more relevant semantics than just being able to try to infer something based on things that we can directly measure, right? And so one of the things that Judea-Pearl advocates for is to perform some of our Bayesian kind of inference, techniques, in a semantic that enables us to actually, directly encode some sort of action or some sort of activity, some sort of perturbation or what he describes as a counterfactual analysis. And so there's certainly, there's a family of computer scientists who are developing techniques to approach questions of this form in this way. But again, for me as a biologist, I think that while these approaches can help us be able to build predictive models that have perhaps very high predictive efficacy, it's not necessarily exactly the same thing as what we mean when we say we want to discover or we want to understand a causal mechanism. And so there's this really beautiful perspective piece written by Michael Levin in Nature Reviews Molecular Cell Biology, I think last year, where in his abstract he really says, what does it mean to say that event X caused outcome Y, either in our experiments or in this case of what we're talking about these couple of days of infectious disease, for whatever sort of disease phenotype that we're trying to study. And that really, answering these types of questions of this form are really fundamental goals for those of us who are actually basic scientists. And so I think in order to answer this question, I think we need to have a common consensus or a common understanding of what it is that what we mean when we say the word mechanism. And many times when we look in, say, our favorite journal, we see mechanisms described or depicted in the form of variations, various network models where perhaps they go across scales, or maybe they stay at one specific level abstraction, and you have many different genes or protein products or metabolites, et cetera. And what's described is that maybe this subnetwork is what's the difference between two different phenotypes that we can see. But I think that there's value in maybe turning to the dictionary and maybe kind of unpacking further what mechanisms really are in a practical level, the way that we think about them in common language and usage. And so if you were to look in a dictionary, what you see is that, of course, you see terms that are very familiar with how we use the phrase mechanism, right? So for instance, a mechanism is composed of pieces, is composed of parts, and that the organization of these parts allow us to make predictions. But there are several other traits which I think are relevant for us who are trying to understand these sort of things. And one of these is that mechanisms are not only a collection or a count of the number of biological objects, right? A mechanism is not X, 3 units of this node and 5 units of that node, right, et cetera. But rather that mechanisms are these causal chains or series of events where something happens, it creates a new condition that drives something else that happens, et cetera, which eventually elicits or enables us to, enables the system to manifest the phenomena that in this case maybe we deem as like the infection state. And second, that that mechanisms, at least in the dictionary, ought to be constructed in a way in which that they're actually derived from first principles. And really, it's the first principles component that the trait of that these things are being built from first principles is what enables the predictive ability. In contrast to say many of the techniques that are really popular right now in machine learning, where purely data-driven approaches, there really isn't a first principles construction. And so how do biologists tackle this? Well, I think for most of us, since we were very young, we were taught to try to answer questions about mechanism this way, where we use what's called the scientific method, right? We see something interesting, we ask a question, we make a hypothesis, and then most of the effort in science is number one, trying to define a very, very specific hypothesis that is testable, and then number two, design an experiment that has very, very good controls to be able to test those directly, right? And this kind of approach for testing hypotheses and generating knowledge and kind of building upon what we know about the natural world, right? This style of thinking is something that became popular, or was initiated in the 1600s by this piece by Francis Bacon. Now, for individual cell phenotypes, this is something that's pretty easy, right? I mean, especially for those of you who trained as bacterial geneticists, right? There are some phenomena that are very tractable in this way. But when we're talking about more complex phenomena, such as a certain disease state, especially things that maybe come from idiopathic kind of circumstances, what makes the scientific method difficult is that biology is incredibly complex, and that many of the features or many of the systems that we're trying to study aren't insulated and isolated from each other, but they're tightly, they're not only complex on their own, but they're also deeply cross-networked with other components of a cell or human physiology. And so what's needed are new approaches that can enable us to learn mechanisms that don't rely on kind of this low throughput or kind of methodology where we're constrained to looking at one thing at a time, but things that enable, tools and approaches that enable us to really tackle these questions at scale. And so, in Levin's perspective piece, what he advocates for is perhaps, biologists, we can draw upon some of the tools that have been developed by physicists and people from other domains, including activities such as network science or like Teresa was describing earlier, like mathematical modeling, to really kind of inform and enrich, the biologist's pursuit of kind of hypothesis-driven understanding of a system. And so that leads me to kind of our topic for today, right? Which is, what are ways that we can use machine learning to really advance the ways in which we do causal mechanistic discovery in biology. Now today, I think that we're really privileged that we live in a time where we have a tremendous amount of experimental tools available to us to for instance, do high throughput characterization, as well as computational tools and computational power to analyze these things intensely. And I say, today, like probably the two of the most common conventional approaches for tackling these kinds of questions, are going to be depicted here. So one of these, which I think most of you are really familiar, is what I would call a purely, informatics-driven approach, where, at some stage there's a large-scale data collection, right? And this can either be something like, sequencing or some sort of mass spectrometry-based, quantification. And then we define some sort of subjective heuristic for what we consider to be interesting in the data collection, right? And that might be something like a certain p-value, statistical cutoff, or something differential between a case and control, or it might be a certain effect size. And then what we do is we go find our bioinformatic friends, and we say, given that set of hits, help me figure out what pathways are enriched in these things so I can go do an experiment. Now the challenging thing is that heuristically that for many large-scale data collections, and this is true for even things like drug discovery, biochemical screening, that a typical hit rate's about 1 to 3%. And so the consequence of that is that we have a subjective standard that we define, and then we get very, very little actually, I guess, interesting or actionable information from that data collection. And then the consequence of this is that when we try to do the downstream analysis, that we encounter these statistical power kind of limitations or bottlenecks. And usually what then happens is you do maybe say your ingenuity pathway analysis and you get some very, very vague term like cell proliferation or migration or something that is not really directly actionable for an experimental biologist. In contrast, kind of a family of approaches, and this is what we've talked about a lot today, has been, or these past couple of days, has been what I would describe as purely data-driven approaches, where we're asking computer to find or define some sort of association between the things that we can measure biologically in our different clinical or cellular phenotypes. But as I think everybody in this room is familiar, the challenge here is that these relationships are often encoded in these mechanistically opaque black boxes, where whatever relationships are being defined also are not experimentally actionable. So we cannot perform hypothesis testing. And so what we did is in a very recent paper, what we did is we proposed that perhaps a new way, an alternative approach to using machine learning to kind of guide our discovery of new mechanisms is one in which we leverage some tools, for instance, from network biology in order to augment the machine learning activity. And we call this, we try to be cute, we call this white box machine learning to contrast with what typical black box approaches. And to kind of summarize like the conceptual framework here, what we did is, at first there was a data collection step. And then what we do is we use, mathematical models, mechanistically, and, curated network models, to perform simulations to represent the data that we had in our large-scale data collection. In this case, it was a biochemical screen. And then what we did is we coupled that directly to some machine learning and asked the computer to identify features that were inside the network that were responsible for the phenotypes that we were measuring. And I'll explain that, and I'll show you what we did in the next few slides. We applied this problem to a problem that in the Collins lab, something that we're very interested in, and this is trying to ask this question or to gain better understanding to the mechanisms of action for antibiotics and what really gives rise to variability in antibiotic treatment responses. And so I think everybody probably recognizes, I mean, you guys are all clinicians, so you guys know what antibiotics are, that most antibiotics that we use, the way that we conventionally think about them is that what they do is they target, they inhibit machineries, cellular machineries that are important and essential for cellular replication. And kind of, I think, what's then a dogma is that if you break an important machine, then the cell will just die. But what we've seen and what I think many other groups have seen is that there are several aspects of bacterial physiology that can not only tune the magnitude of response, but also actively participate in how antibiotics kill bacteria. And this is something that I think for those of you who work with mammalian cells or mammalian systems, I mean, this is very familiar, but I think that for us, we found that this was something that was kind of controversial for some time in trying to understand antibiotic mechanisms of action. And so what we did is, kind of armed with our observations, with observations of many people that metabolism is relevant, can be relevant to how antibiotics kill. We performed this, an experimental screen where what we did was we took E. coli cells grown in minimal media, and we counter-screened them against combinations of antibiotics at different concentrations and about 200 different metabolites that are known to participate in E. coli metabolism. And so what emerges from this biochemical screen is that we have a systematic and unbiased input-output relationship between a focal metabolic perturbation, right, which is induced by us adding metabolite to that well, and some shift in antibiotic susceptibility, which we measured by IC50, or which we quantified as IC50. And what we did was we coupled this empirical mapping of how different metabolic perturbations can affect antibiotic susceptibility with flux balance analysis simulations of metabolism corresponding to each of our individual metabolic perturbations. And we can do this using what are today pretty comprehensive genome scale models of bacterial metabolism. So that what we get is we get a very information dense and rich representation of the metabolic state corresponding to each one of the screening conditions each of the conditions that we perform on our screen. And then what we did was we brought together these empirical antibiotic susceptibility measurements with the simulated metabolic network states and asked the computer to identify for us which reactions inside the network were responsible or could be predictive for some sort, the measurable changes that we had in drug susceptibility. And by virtue of properties such as conservation of mass, right, the reactions which get selected actually turn out to necessarily fall into pathways, right? So if you have a metabolite that's sitting in the media, in order for it to reach whatever effector is causing the change of phenotype, right, each of those metabolic reactions actually has to be linked. And so what happens is that the reactions the computer identifies actually already form pathways, and so we performed a bioinformatic enrichment analysis to define what those pathways are, and those led us directly to experiments that we could validate these hypotheses. And kind of just to kind of compare these against how we typically do biology, I think this overall approach addresses like the fundamental issues that you have with purely informatics-driven approaches, namely because here we're not actually imposing a subjective criteria for what we decide to be interesting data from the screen, right? And so instead of categorically, discarding maybe 95 to 97 to 99% of the data, we get to actually, utilize, all 100% of the data that we have in our screen. And moreover, our network models actually allow us to estimate and predict activities that maybe were not directly included in the screen. For instance, maybe we don't have, certain metabolites available to us to do experiments with. And in relation to kind of the second class of activities, in relation to purely data-driven approaches, this model-driven approach, I think, has some advantages in that, again, because what the computer is doing is selecting features, in this case, reactions that are out of the metabolic network models. And because these models are, manually curated, and we actually know the identity of the enzymes corresponding to, many, if not all, of the reactions that are represented here. This actually enables us to, whatever the mappings are being defined, actually is something that is directly experimentally actionable, right? Because what we can do is you can go directly to the enzyme responsible for that reaction, knock it out, and see what happens. So we did this. So this is just kind of a representation of data that we get from our screen. Very obviously, you can see that between our 3 different classes of antibiotics, you get very different kind of metabolic response profiles. But hopefully, one thing that you can appreciate from the coloring, from the shading, is that if you look across different rows here, which are different individual metabolites,
there are many cases where the color of the shading is very similar, even though the quantitative value may not be. And so what this is suggestive is that although the 3 antibiotics that we tested, have very diverse and distinct targets and different mechanisms of actions, clearly because again they had very different metabolic response profiles, that there must be processes that are shared such that whatever metabolites are responsible for these phenotypes are acting through some sort of shared fundamental aspect of bacterial physiology. So we perform this analysis, based on, because we have all this quantitative data, we can actually compute scores to score the different pathways. And so, these are the things that popped out. Not only did we recover many, pathways that have been previously described in the literature, including our own, for instance, many pathways related to essential carb metabolism, the TCA cycle. But what we also discovered were kind of a novel set of pathways that had never been previously implicated. And this is significant because, as you all know, antibiotics have been around for about 100 years. So we actually know, we've done a lot of work, with antibiotics and in cells such as E. coli. And moreover, from all this quantitative information, we have the sensitivity to resolve qualitative differences between how these pathways act. And some of the different antibiotics that we're using in our screen. So, for instance, you see here that our overall approach predicts that pathways relevant to nucleotide metabolism would have different effects in epicillin and ciprofloxacin, in contrast against gentamicin. Again, because we have all this quantitative information, we can open the box, look at the regression coefficients, ask ourselves where this pattern is occurring, and what we found is that this pattern occurs in the early steps of purine biosynthesis. This leads us to a very actionable, hypothesis that we can test, which is namely that, well, perhaps if we perturb, purine biosynthesis, that we would see this pattern where we would have an opposite phenotype between epicillin and ciprofloxacin versus gentamicin. Indeed, when we did those experiments, that's what we see. So if we do these genetically, for instance, by deleting several early genes in purine biosynthesis, what we see is protection, less killing with epicillin ciprofloxacin, but more killing with gentamicin. If we do this biochemically, for instance, by treating with 6-mercaptopurine, which inhibits PRF, the first step of purine biosynthesis, we see the same qualitative phenotype. If we do this, similarly, if we genetically or biochemically perturb processes that feed into purine biosynthesis, such as the folate cycle, we see the same qualitative phenotype. And very excitingly, if we did the opposite experiment where we try to increase purine biosynthesis activity, for instance, by supplying substrates such as PRPP and glutamine, we see the opposite phenotype. So not only were we really excited that that this actually worked, right? But actually, what we were excited by was that this overall approach where we have an experimental perturbation analysis, right, an unbiased experimental perturbation analysis in the form of a screen, combined with the use of a network model to interpret, and represent that screen in conjunction with the machine learning. It was very sensitively able to give us not only the pathway but also these qualitative differences. And so what I'm going to just do now is just share an example of how I think our vision for how we think that this actually may be clinically useful. So while we may derive a lot of pleasure from just writing papers, it's not papers don't exactly themselves directly benefit the patient. And so we've done some work now to extend this overall approach to diseases that are relevant. And so one of these is tuberculosis. So I think for many of you in the room, you know that one of our first-line drugs for tuberculosis is isoniazid, which works by inhibiting, which first must be activated, from its prodrug form, and then inhibits mycolic acid biosynthesis. And for anybody who's done any work with isoniazid in vitro, typically what you see is this very characteristic, initial killing phenotype, followed by a very rapid development of antibiotic resistance and recovery of the culture. So we did this exact same approach where we took, in this case, H37RV cells. We curated a set of metabolites that are known to be involved in TB metabolism. We used a TB model, et cetera. And what we recovered was this set of metabolic processes that were implicated now very specifically to be relevant to isoniazid. So I think one of the things that's really neat and really cool is that what's highlighted here in red is that we basically recover the target for the drug, right? So again, kind of speaking to the sensitivity of this overall approach. But also what we see is that we recover processes, that are very similar to what I just showed you in E. coli, and so in the vein of thinking about how these kinds of these type of, pursuits for causal mechanism can guide our rational design of new therapies. So one of the things that appears is, again, the enrichment pathways that are relevant to central metabolism. And so, perhaps, one rational, rationally designed therapy might be to combine perturbations to central metabolism along with isoniazid to see if that they can improve treatment. And indeed, we did in some of our in vitro experiments that what we see. So in red here is, again, that very characteristic, recovery due to resistance from isoniazid. What we did is we use subtherapeutic concentrations of, in this case, Q2,3 and bedaquiline. So depicted in blue, you can see that these concentrations themselves don't actually have any sort of lethal effect. But what was really compelling, what was really cool, is that when we combine these two drugs, we saw not only kind of increased efficacy, but in some of our replicates, actually, we got complete sterilization of the culture. So again, I just want to. So I guess I just want to close with kind of a few thoughts on where, how I think this overall approach can really be impactful for the ways in which we, think about, again, the rational design of new therapies, but also, things such as disease prevention. And so, as Sarah described, so I just started, a new job 6 weeks ago. I'm now at the Rutgers, New Jersey Medical School. For those of you who are, trainees in the room, we're recruiting. So, if you're interested in anything that we talked about, please, come talk with me. And kind of one of the components of our research vision is that of course, there's tremendous value in the ways in which people are thinking about using AI for healthcare, today. I mean, and all the wonderful talks that we have these past couple of days, exemplify that. But one of the things that I'm personally very interested in is perhaps, can we, strategies that we can use to very directly learn mechanisms related to either disease pathogenesis or therapeutic efficacy directly from clinical biospecimens. And sort of the proposal here is that, the approach that we describe, that I just described is actually, in principle, from first principles, right, ought to be generalizable. It's agnostic to, what kind of cells that we're using. It should be agnostic to what kind of network models we're using. I gave you an example here with E. coli and metabolism and the phenotype of antibiotic susceptibility. But there's several other phenotypes, there's several other types of cells, there's several other kinds of systems that we can look at. And really, the kind of our vision is to develop integrated approaches, as well as advancing the ways in which we form network simulations, such that one day we can actually build very specific patient-specific versions, representations of say the cellular biology of healthy and disease specimens in a way that kind of in one step may directly lead us to new mechanisms that we can directly validate. And just to give you an example of one of the ways in which we're doing this now now at Rutgers. So this is in collaboration with David Alon, which I think somebody mentioned GeneXpert earlier. He's one of the founders for that technology. The Alon Lab, I think was this last year, 2 years ago, published this really exciting, kind of really interesting paper, where what they did was they examined clinical isolates from this TB study, where there were patients which were given like their normal four regimen therapy and certainly were completely cured of TB, but there was also some fraction which relapsed. And so what the Alon Lab asked was, what is the difference between isolates from the patients which were able to have their TB cured with normal TB therapy versus the ones which had a relapse infection? And what they observed was that if you think again related to KP's talk just now about say drug susceptibility testing, how in practice what we do is we perform some sort of microbial dilution and we define some threshold and we say this threshold is the threshold by which this pathogen is either drug susceptible or which is this where this pathogen is drug resistant. Is that the isolates which came from the relapse patients actually were sub-breakpoint, right? So these were, the MICs that they measured in the experiments were not materially different and would not be above the threshold by which you would call these strains as being drug resistant. And so this would be an example where, of course these are wild isolates, so there are many different genotypic differences, but this is not due to some canonical resistance mechanism where you have a mutation, for instance, in a target. And so we actually very recently just received funding from the NIH, to deploy, this metabolic screening and, white box machine learning approach I just described, along with a few other techniques to kind of build individual metabolic, models and representations for each of these different, for about 60 of these different strains, from this library, and then to use this overall approach to figure out, what might be the metabolic differences that differentiate strains that are able to cure versus ones which, are doomed to relapse. And so with that, I just want to acknowledge several of the people that I've worked with over the years, and on data that I presented today, and much of our funding supporting this work was from either NIH or DARPA or DITRA. Yeah. And so with that, happy to take any questions.